The central goal for the development of the CIBSS technique was getting comprehensive access to the organization and assembly of protein complexes that underlie signal transduction at and across the plasma membrane of various types of tissue and organs.
CIBSS currently provides the highest resolution and versatility for analysis of native protein complexes and their subunit composition, in particular with respect to membrane proteins.
Although the CIBSS technique was developed to analyze complex mixtures of membrane protein assemblies in rodent brain, it can easily be adapted to the analysis of any type of biological sample. One of the key steps of our method is the embedding of the gel piece and its correct alignment to the cutting plane before slicing. Be careful not to tear or to compress the gel, and be sure that it is not tilted to the cutting plane, since this would reduce the resolution of the analysis. Our CIBSS technique has several practical steps that are crucial for good results. It is easier to show how to perform those steps in detail than by just describing them.
To begin this procedure, use a stirring 2-chamber gradient mixer driven by a pump to cast a linear or hyperbolic pore gradient gel. Prepare solutions for the front mixing chamber and reservoir chamber as outlined in the text protocol. Start the stirrer and add 30 microliters of APS and 2.5 microliters of TMED to the solution in the front chamber. Then, start the pump and open the front valve. After approximately 1 minute, add 90 microliters of APS and 5 microliters of TMED to the reservoir chamber and open the chamber connection.